Hello, everybody. I am Juan Caicedo, and it's my great pleasure to present our work today, predicting compound activity from phenotypic profiles and chemical structures. This work was led by Tim Becker and Kevin Yang in collaboration with researchers at the Broad Institute and MIT. A common use of microscopy images in biology is drug discovery, and at the core of drug discovery is the ability to predict if compounds exhibit a specific activity. In drug discovery projects, institutions may have hundreds of thousands or even millions of chemicals available, and the goal is to identify which chemicals are potential treatments for the many diseases that still need to be cured. Typically, experts for each disease design an assay where the readout reveals at least one property of interest to treat such disease. These assays can be biochemical, where we look for protein binding activity, or they can be cell-based, where we look for specific cell responses. Even with automation, it's tremendously expensive to carry out all assays for all chemicals in every disease. That's why we have a sparse knowledge of which compounds have which activity. We only know the results for a few of them because we can't test them all. Computational approaches that predict the results of assays using existing information would be very powerful to expand our knowledge about compounds. In 2018, a team of researchers demonstrated that it is possible to repurpose pre-existing imaging screens to infer assay outcomes from seemingly unrelated information. A 3-channel glucocorticoid receptor high-throughput imaging assay was used to make predictions in an oncology project and a central nervous system project. They had a few assays of interest, with the corresponding results for a few compounds. So, they took images of cells treated with the compounds and transformed them into morphological profiles where we can measure cell state in an affordable way. Then, a machine learning model was used to complete the missing values in the assay matrix, and from those predictions, only the highly accurate predictors are preserved for selecting promising drugs that can be tested in vitro. The results show that this approach can help improve hit rates up to 250 times, making the use of resources much more efficient and the drug discovery process more effective. In addition, advances in the analysis of chemical structures have recently allowed researchers to identify effective drugs just by looking at the molecule structure using deep learning. With these methods, we can now predict whether a molecule would result in a positive or negative readout for a given assay without physically testing it. The analysis of chemical structures can result in the exploration of a very large space of chemical matter, allowing us to discover structurally diverse molecules. Using this approach, our collaborators at MIT discovered a novel antibiotic called halazine, which exhibits efficacy among a broad spectrum of bacterial infections. So, here we ask the question, how can we use the tools for chemical structure analysis together with powerful phenotypic profiling for assay prediction? This is the approach that we explored. First, given a compound library, we use ChemProp for chemical structure analysis, and we train models to make predictions in a diverse set of assays. Then, we use cell cultures and perturb them with the compounds to observe their response using imaging, more specifically cell painting. Morphological features can be added to the machine learning model to facilitate predictions. Finally, we also used gene expression as an additional phenotypic profiling method to be analyzed by the machine learning model. With this approach, we wanted to understand how we can obtain better assay predictions for a wide range of compound activities. Notice that as new assays are added to the pool, the imaging and gene expression profiles do not need to be created again. The library of morphological and transcriptional profiles can be reused to train another predictor for a new assay as long as some training examples are collected in the lab. We used a set of 16,000 chemicals, 13,000 for training and 3,000 for holdout for validation. We collected cell painting profiles together with L1000 transcriptional profiles from a publicly available dataset. Next, we collected the results of 376 assays used to test the activity of these compounds from researchers at the Broad Institute who worked in various drug discovery projects. Not all compounds have been tested with all assays. In fact, 92% of the combinations is missing. We have the biological details for only a fraction of those assays. The remaining annotations are confidential. However, we tested the prediction ability of all of them anyways. And the assays with annotations include yeast, fungal, cell-based, bacterial, and biochemical assays, representing a wide spectrum of biological activity. Finally, we use the ChemProp software to train the deep learning models used to make predictions in all of our experiments. And here are the results. When we test single modalities for predicting the 376 assays, we found that they show diversity of prediction strengths. The numbers in the Venn diagrams indicate the number of assays predicted with high accuracy. Here, MO means morphology, CS is chemical structures, and GE is gene expression. Morphological profiles are the best individual modality for assay prediction, yielding highly accurate results for 67 assays in total. We combined the profiling modalities and found that it is possible to integrate their strengths. Here we show the assays predicted in common by all four combinations of profiling modalities using late data fusion. Adding morphological profiles to chemical structures improves assay prediction ability. The trained models can predict a diversity of assay types, and they don't seem to be skewed to only predicting cell-based assays. We also found that chemical structures, morphology, and gene expression profiles provide complementary information for prediction. And if we take the best predictor for each assay from all the candidate models, we can predict 115 assays in total, about a third of all the assays in our study. In general, assay predictors can improve hit rates. In this plot, each point is an assay, and the x-axis shows the baseline hit rate observed in the lab, while the y-axis shows the falls of improvement if a machine learning predictor is used instead. Purple points are highly accurate predictors, which can usually improve performance up to the theoretical limit. And for assays with rare hits, the predictors can offer high efficiency as well. And finally, the performance of predictors does not correlate with the size of the training set in our experiments. Here, each point is an assay predictor, and the x-axis shows the number of training examples, while the y-axis shows accuracy. High accuracy can be reached with a few training examples, and having lots of training examples sometimes cannot help to obtain higher accuracy. We hypothesize that predictability has more to do with the biology and the assay itself. So, in conclusion, morphological profiles are the best individual modality for assay prediction. Adding morphological profiles to chemical structures improves assay prediction. Models can predict a diversity of assays, and modalities are complementary to each other. About a third of all the assays in our study can be predicted computationally. With that, I want to thank Tim, Kevin, Shantanu, Tommy, Regina, and Anne for their contributions to this project, as well as our many collaborators at the Broad Institute and MIT. Thank you all for your attention.